Welcome everyone to another lecture. So, if you have watched the previous lectures, we talked about DNA replication, we talked about DNA structure and function, and also now we are here. We also talked lastly about DNA repair, and the different types of repair, sources of DNA damage, endogenous and exogenous sources. Now we're going to be looking at the different types of DNA damage, DNA repair. So we're going to be looking at base excision repair, nucleotide excision repair, and non-homologous end-joining and homologous recombination. But I'm going to be starting this video with base excision repair. So we have base excision. All right. Okay. So like we said in the previous video, if you have not watched my previous videos, please make sure you do them, because you need to build on previous knowledge for you to actually understand this thing, because we are going on a journey. All right. So base excision repair. Base excision repair simply talks about you actually repairing a single-stranded break, or in this case, a kind of a mismatch or a spontaneous change that has occurred at the nitrogenous base. Now, in base excision repair, what we are actually repairing is an error in the nitrogenous base, whether it could be as a result of deamination. So when deamination occurs, deamination simply means when the amino group is being removed and ammonia, NH3, is being released. So that's what deamination talks about. And when such a thing occurs within the cell, the repair mechanism of the cell is going to be a base excision repair. It just has to do with removing a base. And I'm going to show you how it goes. So, like I said, deamination, just to give you some things on deamination. Now, when adenine is being deaminated, it produces hypoxanthine. When guanine gets deaminated, it produces xanthine. When cytosine gets deaminated, it produces uracil. And then you need to know that thymine doesn't get deaminated. It doesn't get deaminated because there's no amino group there. So now we want to look at a sequence and use that to explain what we are talking about here. So take this sequence. If you had 5', of course, I think by now you should know that DNA synthesis occurs from 5' to 3' direction. So it's bidirectional, so the opposite strand will have the opposite in the opposite direction. So we have 5', we have a sequence like this, GCTA, all right. So from what we have explained in previous videos, we know that adenine binds to thymine and cytosine binds to guanine, right? And that's because a purine has to bind to a pyrimidine, isn't it? A purine binds to a pyrimidine. So these both are pyrimidines, and then these both are purines. So there's a problem here, because in first of all, this actually is a DNA, the deoxyribonucleic acid, nucleic DNA molecule. And then you know that the difference between RNA and DNA is that in DNA we have thymine, while in the RNA you have uracil. So this tells us that there's actually a problem here. Let me use a different color. There's actually a problem here. The cell has a system which helps to kind of like check for such errors. So it consists of a battery of enzymes known as DNA so DNA glycosylases, and there are actually six types. So for the different nucleotides and for the different situations that may occur and lead to lead to this repair. So in this situation, let's not forget that we have hydrogen bonds. Hydrogen bonds. And you know the number of hydrogen bonds between guanine and cytosine is actually 3. Okay, sorry. And then between adenine and thymine is actually 2. But I'm not going to show that here. So we have these hydrogen bonds, and just for clarity, we need to know that the bases will be within this place here. So let me change it so that it may make more sense to us. So this is the sugar phosphate backbone here. And then these are the bases here, forming the bonds together. So the first thing we have here is that a certain enzyme, since this is uracil, you have uracil glycosidase. So uracil glycosidase comes, and the first thing it does is that it removes the base, it nicks out the base. So we have the base removed here. The base is removed. So off goes uracil. It goes away. And after this, the next enzyme is, so this nitrogenous base, this DNA, for example, doesn't have the uracil here. So the next is what we call endonucleases. And one thing you know about endonucleases is that they cut within the nucleotide chain. But for exonucleases, they cut at the ends, not within. So, and it's an endonuclease. And the meaning of AP is apyridinic and apurinic AP. So what it does is simply this. It breaks the phosphodiester bonds here. Breaks the phosphodiester bonds. Together with phosphodiesterase. It breaks the phosphodiester bond here. And then leads to the removal of this sugar phosphate backbone. You know, there are phosphodiester bonds in between them. So they are broken and it's removed. Excised. That's the right word. Then the next thing we have is that, you know, there has to be synthesis. Therefore, you should know the enzyme that is responsible for synthesis of DNA, which is DNA polymerase. So DNA polymerase comes. Don't forget 5' to 3'. There's going to be an OH here, so it uses that as one of the substrates for the reaction. It comes here and it catalyzes the synthesis. So after DNA polymerase catalyzes synthesis, next thing we have here is that there is an enzyme that has to join them, which is DNA ligase. So DNA ligase helps to join the break of this DNA synthesis. And of course, you know, when the synthesis occurs, there will be the replacement of the correct nucleotide. I missed that. I forgot to put that. Of the correct nucleotide during DNA synthesis. So, and that's what DNA base excision repairs are about. So the next video will be on nucleotide excision repair. Nucleotide excision repair.